Welcome to Roasted Academy. So today let's talk about the spermatogenesis, which is the formation of the mature sperms. So when you are approaching embryology, I always advise to say that first you need to look at gametogenesis, which is the formation of the gametes, sperms as well as the ovum. So you go way back in the female, it's oogenesis. In the males, spermatogenesis. So when you finish that, you look at from when these gametes are going to fuse together. That's fertilization, right? Then you start the weeks of development. Week 1 all the way up to when the fetus gets implanted into the endometrium. And then you proceed with the rest of the weeks, right? So you don't just start from week 1, no, you start from way back when the gametes have to be produced. So what is spermatogenesis? Spermatogenesis is a process by which sperms are produced in the testes, starting with the spermatogonia, which are stem cells located in the seminiferous tubules. So we have these cells, spermatogonia, which came from the yolk sac, migrated from the yolk sac all the way up to the testes. So they start proliferating, undergoing divisions, which is mitosis. Then when they reach here, they become spermatogonia. So these are diploid cells that undergo mitotic division. Some remain as stem cells, while others will differentiate into primary spermatocytes. So primary spermatocytes, these diploid cells will undergo meiosis 1 to form secondary spermatocytes. So secondary spermatocytes, these are haploid. When we say 2n, we simply mean that the number of chromosomes is 20, that's n, 23. So when you multiply 2 times 23, that means it's 46. Okay. So sex cells are the only cells that contain haploid number of chromosomes, which is 23. So normally we have 46 chromosomes in the cells. But what we see is that these cells, when they undergo meiosis 1, they become secondary spermatocytes and they have haploid number of the chromosomes. Then the secondary spermatocytes, these cells will also undergo meiosis 2. They will produce spermatids. So that's when we have got the formation of spermatids, which are also haploid cells. And these are the precursors of the mature sperm cells, the spermatids. So let's go to the next one, spermatogenesis. So this will convert now the spermatid to spermatozoa. During spermatogenesis, these spermatids undergo significant changes to become mature spermatozoa, which is sperm. So formation of a sperm structure, the spermatid elongates and develops key structures. So now we have the head on the sperm. We've got the head, we have the acrosome, we've got the midpiece, we've got the tail. You need to know how to draw the sperm, the structure of the sperm, and also know what it's containing. The head contains the nucleus with the genetic material. Then we've got an acrosome, a cape-like structure on the head, which contains enzymes essential for fertilization. We also have the midpiece, where the mitochondria is, which provides the energy for the movement of the sperm itself. Then we've got the flagella, which is a long whip-like structure and allows the sperm to swim. So that's the second one. Let's go to the third step. Sperm maturation in the epididymis. After spermatogenesis, sperms are released into the epididymis, where they undergo maturation. So we've got these processes like decapacitation. In the epididymis, sperm undergo decapacitation where the acrosome cape is partially or completely removed. This is to prevent the premature activation of the acrosome, which is important for ensuring that the sperm is not activated before reaching the egg. So we don't need the sperm to be activated at this point, right? So decapacitation allows the sperm not to be activated because it's not yet in the female reproductive tract, right? Then maturation, the sperm also gain the ability to swim during this process now, becoming motile and capable of moving through the female reproductive tract to reach the egg. So now let's go to the other one. Mature sperm are stored in the vas deferens until when there's ejaculation. So take note of that point. Vas deferens, that's where uh, the storage of the sperm until there is ejaculation. During ejaculation, sperm are mixed with the seminal fluid from the prostate and the seminal vesicles to form semen, which is expelled now through the urethra. So capacitation is the final step. Maturation, it occurs just after ejaculation when sperm enter the female reproductive tract. What happens during capacitation? There is now the sperms will gain motility. Motility increases, allowing for more vigorous swimming and better invagination navigation towards the egg. Acrosomal reaction readiness. Capacitation makes the sperm ready to undergo the acrosome reaction when it encounters the egg. This includes the release of enzymes from the acrosome that will help the sperm penetrate the egg's protective layer. So what's the importance of capacitation in case you come across a multiple choice question? This step is critical because it ensures that the sperm is capable of fertilizing the egg. Without capacitation, sperm would not be able to properly undergo acrosomal reaction and fertilize the egg. So once the sperms have been capacitated, okay, they encounter the egg in the fallopian tube. So we've got those different parts of the fallopian tube, which we'll talk about, right? And you need to know which side is where does exact fertilization takes place from the ampulla, right? The sperm releases enzymes from the acrosome to break down the egg's protective layer, which is the zona pellucida, allowing the sperm to penetrate the egg and deliver its genetic material. This fusion of genetic material from the sperm and the egg will now form a zygote, which begins to develop and to divide and develop into an embryo. So in the next one, we'll talk about oogenesis. Oh, we've already talked about oogenesis. We now have to move to fertilization. That's how we move. Move from oogenesis, spermatogenesis, then fertilization. Fertilization and its results. Right? Thank you so much.